much. I'm really excited to be here to tell some of our lab's work. So my lab's long-term goal is to build predictive models that link cellular states and RNA sequences to the endogenous RNA's translation efficiency. And to be able to do that, we really need two things. First, we need quantitative measurements of translation efficiency across many different cellular contexts. And second, we need computational methods to distill these measurements into predictive models. So how do we actually make these measurements of translation in a transcriptome-wide level? So we've already heard about this technique, ribosome profiling. And the really key idea of this technique is something that we've known for probably 25 years. And that is ribosomes are stable molecular machines, and they can actually protect the RNAs that they're translating from digestion. And what we can actually do is isolate these protected fragments, and by high throughput sequencing of that, we can get a snapshot of ongoing translation in cells. So since its development, this has been a really popular method, yet unlike standard RNA sequencing, one of the bigger limitations is the need for really large input. So we actually typically need millions of cells to execute this technique, which has really limited its application to cell lines and other large organs. So my lab and others in the last few years have been really coming up with methods that has been pushing this technology to make measurements in single cells and other little precious low input sample types. So this will undoubtedly really increase the types of cells which we will generate these kinds of measurements. But already there have been more than 10,000 experiments that have applied this technique to measure translation in different biological contexts. So what we really wanted to do is, how can we really make all of this data reusable for integrative analysis beyond the original publications that reported these studies for the first time? So to be able to do that, perhaps the easy part is really having a computational ecosystem that can really scale to such large numbers of data sets. And this is something that we've been doing over the years, which has a key component that's a ribophile that really helps us process large numbers of data sets. But perhaps, as you can imagine, the bigger challenge is really trying to get all the metadata that is required to understand the different experimental variables, the different experimental conditions that were used to generate these data sets. So I just want to show you how difficult this really is. So these are actually two studies from really well-known labs in our field. And these are data sets they've generated and deposited to GEO, a common database for this. But what you'll notice is one of them actually calls this ribosome profiling data RNA-seq. The other calls it other. One of them mentions that it's ribosome profiling at least somewhere. But this one actually includes some of the key experimental variables that are necessary to reprocess this data. So unfortunately, there's a lot of variability and unstructured information that really limits reusing this data at scale. So this is where being at a large undergraduate research university helped. So we've made training videos, and we actually hired many undergraduates, which is led by this team of undergrads. And they actually went through every single one of these studies to be able to validate, the type of the experiment and critically match which ribosome profiling goes with which RNA-seq data and other key experimental variables. So this is really what allowed us to actually reanalyze all the existing ribosome profiling data and create like a resource that will allow us to do these integrative analysis. And today I'm just going to talk about our work in human and mouse data sets. And in particular, these include more than 3,700 ribosome profiling experiments across more than a couple hundred different sources, cell types. So one really useful thing of having all of this data uniformly processed is to actually execute some key quality control across all of these different experiments. So as you might expect, since this is ribosome profiling data, the root footprints should come from coding regions and other really simple key quality metrics. And unfortunately, many data sets fail even these simple QC metrics. And in fact, even though all of this data comes predominantly from published papers, nearly a quarter of them fail to pass these simple QC metrics. So I think this is really important, not just to highlight like some of the issues, but more so if you're to ever get to a point where we integrate these data sets, we really need to focus on the subset of the data sets that are of sufficient quality. Okay, so now that we have this data set, what can we do with this? So one unique thing that we actually have is that we have the matched samples of ribosome profiling and RNA-seq that really allows us to calculate a quantity that is typically called translation efficiency in the literature. But really what this refers to is how many ribosomes there are when you normalize by the amount of mRNA there. And in particular, we've actually developed a compositional data analysis method that relies on a compositional regression model to derive this particular quantity of translation efficiency. So what we thought was, if this way of calculating TE is an improvement over the standard methods, it should really help us improve the correlation of TE to proteomics data. And in fact, that's what we've solved. So across 7 cell lines that are very well characterized with proteomics to measure protein abundance, if you compare the standard method, which basically just takes a ratio of these counts as a metric of TE, this compositional data analysis method really improves the correlation to proteomics, suggesting that we are making an improvement there. And the next thing that we really wanted to see is that if you look at across different cell types now, that there is quite a bit of variation in translation efficiency of these endogenous genes across these different contexts. So what this data allows us to do is really start to really understand the relationships across conditions of translation efficiency. So in other words, for each gene, what we have is for each of the cell types and conditions, their respective translation efficiency. And this allows us to really define a concept which we call translation efficiency covariation. So the pattern of similarity across cell types. So this is very analogous to RNA co-expression, if you're already familiar with that. So how does this actually look like? So here in this heat map, each of these is a particular cell type and the translation efficiency in that cell type for 3 genes. And what you see is that these particular 3 genes have actually very similar profiles of TE across cell types. And you might recognize these as actually components of the ribosomal small subunit. And this is really saying that their translation is coordinated across cell types. Whereas another gene, hexokinase, which is related to glycolysis, is not really showing a similar pattern. And we can really now quantify these relationships between genes of which ones show similar patterns of translation across cell types versus not. So is this concept, important for cells? And one way to think about it is to really think about its conservation. So if you actually pair genes randomly and calculate translation efficiency covariation, you notice that that's basically centered around 0, whereas for real genes, these quantities are very much correlated, suggesting at least for some pairs of homologous genes, the fact that their translation efficiencies are coordinated across conditions is conserved. So what else can we do with this idea? So one thing we have known for a long time is that genes that have similar levels of RNA expression, so those that are co-expressed at the RNA level, tend to actually have similar biological functions as opposed to those that do not have similar patterns. However, is this true for translation? Do genes that have similar functions tend to have similar patterns of translational efficiency across conditions? So can we really look at that question? And perhaps this is important because we can also start to ask what are some regulatory mechanisms that enable such coordination of translation across conditions. So to be able to do that, we can actually just do a very simple exercise and ask if the genes that are annotated with a particular function are showing similar patterns, they will be having a high score, whereas if not, they will be basically what you would expect by random chance. And indeed, what we saw was translation efficiency covariation is actually almost as informative as RNA co-expression in terms of indicating shared functions between genes. And what's really interesting is that there are actually specific types of functions where translation efficiency coordination is much more informative than RNA co-expression. So for example, genes involved in MAP kinase activity are not really co-expressed at the RNA level, yet their translation is coordinated across the same cell types. And we can really start to quantify this and use this actually to start to make even new predictions about function. And in fact, one really interesting finding that we found is we identified a particular gene that has an LRRC domain, which showed very high similarity in terms of translation with almost all of the components of the glycolysis pathway. And given that these proteins typically are parts of protein-protein interactions, what we actually did was to evaluate potential protein-protein interactions between this new gene of unknown function and the different glycolysis factors. And surprisingly, what we found is that it actually has a very strong predicted binding with transcription factor that regulates all these components of glycolysis. So with that, just want to conclude. So what we have really is the largest resource of uniformly processed quantification of translation efficiency. And today I told you about translation efficiency covariation, but as you can imagine, we've been using this resource for many other purposes as well. And we really feel that it will be very helpful for the community in general. And in particular, in today's talk, I hope to be able to show you that this concept of covariation is conserved between humans and mouse and is actually useful in predicting protein function beyond what can be seen from just RNA co-expression. And with that, I would like to thank the people in my lab. And if anyone's interested in these topics and looking for a postdoc, we're always very excited to consider new applications. So thank you very much.
Any questions for John?
Thanks, very nice talk. So the translation efficiency available in the database, and are these calculated per isoform or per gene?
Yeah, great question. So in this particular case, as we've heard, most of the genes have really one dominant isoform, and we really focused on that. And that's purely because of the challenge of assigning these short ribosome footprints in an isoform-specific manner. And in particular, even though it's possible, it's obviously not as easy as for RNA-seq. And that creates a systematic bias where you have much better estimates of isoform abundance for RNA, not as well for ribosome occupancy, that leads to really biased estimates of their TE. So for that reason, we are focusing at the gene level using the dominant isoform per each gene.
Any plans to attempt the isoform-based?
So not at this point, but yeah, not at this point.
Thanks.
Next.
So I have another question. You mentioned that there's quite a few data sets that actually fail, your quality controls. Are there certain patterns that arise, like what typically goes wrong? Because that might be, you shouldn't maybe point out who, but point out what goes wrong so that people in the future pay attention to.
Yeah, definitely. I think we've looked at a few common patterns, so one actually idea we had was maybe like data sets from earlier on would have lower quality, and over time maybe things have improved. So we've tried to correlate with fraction by time, and it's actually turned out to be the exact opposite. So it turns out that data sets from earlier were higher quality, and the quality keeps getting worse, unfortunately. So I think the issue is, like, in earlier days, people were more critical of the data quality, and now we are reaching a point where people take it for granted that this is a standard method, which causes a little bit concerning from that perspective. And there are other things that we analyzed. For example, these are very, experimental details, maybe not super of interest to people here, but for example, what enzyme is used to generate the data set, and those are also parts in our paper. We describe some of those as well.
Actually be interesting to check other types of genomics experiments as well, because I mean this idea that people start taking for granted, could apply.
Be true. Exactly.
Okay, yeah, thanks for the nice presentation. I was wondering what you think the difference in the translation is caused by. For example, do you think it's a difference in the RNA sequence, like codon preference, or is there any other regulatory?
Thanks so much. I mean, so that's actually another project in the lab. Like, given this resource, we feel that it's really a great resource to build, again, machine learning models that take into account now the endogenous RNA as opposed to a reporter construct, and link that to cell type specific translation efficiency. So, and we have actually a work that focuses on that. And it actually yields many different features, as you can imagine. So some of them are sequence intrinsic. So these are really, given the sequence, there's a difference in a cell type specific manner, but others are sequence extrinsic. So these could be things like, what is the protein complement of that cell that results in translation differences as well. But I guess the short answer is it's a combination of both, and that's definitely of great interest to us.
Hi. So a really short question. You said you have hired many students and graduates to go over the data. So you only filtered for ribo-seq associated things, and all of the rest you just kind of say it's not, or did you also try to classify that as well?
Yeah, thanks for the question. So it's actually a combination of computational with manual curation. So the computational part was really trying to come up with actually in broader sets, anything that could potentially be ribosome profiling automatically. And then when the students were going through it, they were playing around and saying whether it is. And actually, we have those other, potential translation measurements, but not ribo-seq in there, but we currently discarded them. So we have not worked with it, but they are labeled. So we are not planning on releasing that information, but if you're interested, we'll be happy to share.
I mean, that could be
useful for other things.
Even for other experiments, right?
Exactly.
So yeah.
Yeah. Thanks.